Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Enhancing pDNA Production for Gene Therapy with BioNTech. This is the first session in today's virtual event, Biopharmaceutical Analytics Unveiled, Expert Insights and Customer Success Stories. I'm Roisin McGuigan, an editor at BioInsights, and joining me today I have Blaj Balakar and Ahmet Hilmi Tekin, who will discuss the growing demand for plasmid DNA in gene therapy and innovations to streamline pDNA production and analysis. After the presentations, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the left side of your screen, and we'll try to get to them during the session. Now, I'd like to introduce our presenters. Blaj has a BSc in biochemistry and a PhD in nucleic acid biophysics, and is the PATfix product manager at Sartorius BIA Separations, leveraging his expertise to drive the platform's success. Ahmet has a background in microbiology and synthetic microbiology, and is now a senior operational scientist at BioNTech, focusing on process development and optimization for microbial plasmid manufacturing. So without further ado, I'll now hand over to Blaj to kick off today's first presentation.
Thanks, Roisin, and welcome everybody to today's presentation on how to enhance pDNA production for gene therapies. So as everybody probably knows, the global demand for pDNA or pDNA alternative has grown significantly in the past few years, especially due to the pandemic efforts at the start of 2020 and 2021. And accordingly, the scale and production has increased in volume, not just in scope. pDNA is basically an enabling product and is critical in the production of mRNA, AAVs, and other gene therapy therapeutics. So increasing the yield and purity in the production of pDNA is a vital step in meeting such increased demand. That's why today, and for the past few years, we've started offering our analytical solution to help you monitor your production and purification process at line to support you basically with rapid process development and optimization and provide reliable in-process control. So if we look at the pDNA purification workflow, there's quite a few steps in this, and once you start scaling up to higher and higher volumes, some of these can get very challenging, especially, I would say, steps, pre-capture steps. So in this case, it would be pre-simultus DAE capture steps, as shown here in an example purification process. Now, using the PATfix pDNA platform, you can analyze in-process samples very easily and with a guide on how to prepare them to basically give you results on how much content, pDNA content you have. Now, this is challenging because pDNA represents less than 3% of the cleared lysate in preparation. And not only is it a very small fraction, it's also very difficult because some of the contaminants that we need to get rid of in the process are very similar to the pDNA itself, such as negatively charged RNA, genomic DNA, and endotoxin, and also can be similar in size, such as genomic DNA fragments, and in hydrophobicity, like endotoxin. So scalability of lysis and method and process to large volumes is difficult because pDNA is also shear sensitive and pH sensitive, which can create the open circle isoform, which is usually an unwanted byproduct. Now, analyzing these impurities as well as pDNA in your sample in such early complex samples is a bit difficult. And that's because the nucleic acids can actually change their absorbance based on the matrix effect, which is basically dependent on salt concentration and pH of the solution. While on the other hand, dyes that can be selective for pDNA or mRNA or RNA impurity, for instance, can have off-target binding effects that can skew results. And both of these errors can contribute to erroneous readouts that can lead to wrong conclusions. So analytical chromatography is the solution here, where we do separation before we do quantification. And this is an example shown here on screen, where if we take this crude lysate and analyze it on the PATfix platform, we can see on the graph that proteins and other impurities elute first, followed by RNA, followed by open circular pDNA and supercoiled pDNA, which is our desired product. In this way, it's basically very much easier to quantify the pDNA content from the very start and to optimize these early pre-capture steps. In this fashion, what PATfix allows you to do is to collect samples as they're coming, collect process samples as you're moving along, and immediately analyze them and have the results in the same day, instead of collecting them and sending them off to another laboratory where they can be analyzed, which would basically then lead to delays, slowing down your overall process. Now, not only that, when we developed this PATfix platform, we've made sure to make it, first of all, very robust. So we've tested it on many, many, many samples and made sure that the method performs as expected. And it can operate with multiple detectors. And the method itself is also validated according to FDA and EMA guidelines. PATfix comes not just with the hardware, but also with the columns, the method, and the software, everything that is required, including a standard to allow you absolute quantification. Now, to tell us a little bit more on how this looks like in the hands of a user, I'm going to hand off to my colleague, Nate, who's going to tell us a little bit more about it.
The market for gene therapies continues to grow, and the need for high-quality plasmid DNA is increasing. My name is Nate Paulin, and I'm delighted to welcome you to this product demonstration on the PATfix pDNA analytical platform. Many companies are looking to insource pDNA production and purification, usually as a key starting raw material for subsequent viral vector and mRNA production and purification. Our extensive experience of working with numerous companies involved in the production of plasmid DNA for over 20 years has enabled us to gain valuable insights into the key challenges our clients face in their plasmid DNA production processes. One of the most common issues we face is the inability to perform analysis on lysate samples in the pre-capture step of plasmid DNA production due to the complexity of the lysate samples. Many are familiar with lab-scale plasmid isolation via spin column kits. However, knowledge gained from lab kits is difficult to implement in a large-scale pDNA purification process. As a result, our clients have been unable to obtain crucial data regarding the plasmid DNA content within the bacterial cells. Furthermore, the absence of this information has hindered efforts to optimize the lysis process, which is recognized as a pivotal step in the recovery of plasmid DNA from bacterial cells and in maintaining its purity. Another significant challenge we have identified through our work with a number of customers is the lack of effective quantification techniques for determining pDNA concentration. The most commonly used method for determining pDNA concentration is nanodrop. While it is a rapid spectroscopic method that requires a minimal amount of sample, it can also lead to inaccurate measurements and errors. Firstly, nanodrop is unable to distinguish between the different nucleic acid commonly present in pDNA-related samples, such as host cell RNA and DNA. It can also give false quantification results due to the presence of many impurities that can absorb UV light. This can lead to errors in the recovery of the purification process. Second drawback of this type of spectroscopic method for nucleic acid quantification is the influence of pH and conductivity of the matrix in which the pDNA concentration is determined. This phenomenon can also contribute to inaccurate determination of pDNA concentration. A further limitation of this technique is its inability to distinguish between different pDNA isoforms. The objective at the end of the purification process is typically to obtain a single pDNA isoform. The proportion of individual pDNA isoform may vary throughout the purification process due to the inherent instability of pDNA. A lack of knowledge regarding the isoform composition of pDNA and the presence of contaminants, such as genomic DNA, may result in complication when utilizing the pDNA in subsequent processes. An existing solution to this issue is the utilization of electrophoretic techniques such as agarose gel electrophoresis and capillary gel electrophoresis. However, the primary limitation of agarose gel electrophoresis is its inability to quantify pDNA isoforms, while the use of capillary electrophoresis is challenging and requires a great deal of experience. The shortcomings of conventional pDNA analytical techniques prompt us to develop a streamlined, single-method chromatographic solution for pDNA characterization. The main objectives we sought to achieve during the development of a method for pDNA characterization were the ability to robustly analyze all samples involved in the pDNA process, the capability to distinguish between all possible pDNA isoforms, and the capacity to quantify and monitor all contaminants present in different pDNA samples in addition to individual pDNA isoform quantification. Furthermore, we aim to ensure that the analytical method is straightforward in terms of sample preparation, processing, and interpreting the results. We are pleased to present our solution, the chromatographic PATfix pDNA analytical platform, which has been specifically designed for the analysis of pDNA samples at all stages of the pDNA process. The process of developing the method commenced with the selection of an appropriate chromatographic medium. We identify a solution in the use of a weak anion exchanger CMAC pDNA column with 1.4 micrometer channels, which we produce in-house. This column enables the separation of all essential pDNA isoforms, the differentiation of pDNA from other impurities in the sample, and the optimal recovery of pDNA from the column with no carryover. It is compatible with up to 4 molar salt buffers and 1 molar sodium hydroxide for sanitizing the column. Once the appropriate chromatographic medium had been identified, we proceeded with the optimization of buffer composition and gradients. Our objective was to optimize the method in order to achieve the best possible resolution between pDNA isoforms and other contaminants, to provide the best possible recovery and to minimize sample carryover. The objective was to ensure that the method could be applied to all types of samples encountered in the pDNA production process, from host cell lysates to purified pDNA samples. The full potential of the PATfix pDNA method is revealed in lyse host cell samples. The process effectively separates all contaminants, proteins, host cell debris, host cell RNA, and host cell DNA from target pDNA isoform. The separation of pDNA is also achieved, enabling the quantity of pDNA to be determined throughout the entire production. Moreover, this method enables the tracking of contaminants throughout the entire purification process. This analytical method is also able to distinguish between different pDNA isoforms. In this chromatogram, the separation of the 3 pDNA isoforms is demonstrated. The 3 isoforms are clearly distinguishable from one another. This feature of the method is particularly useful for monitoring and optimizing the linearization reaction. Once developed, the pDNA analytical platform was tested on a wide variety of pDNA samples, including small and large plasmids, plasmids for IVT reaction, and nanoplasmids. However, we encountered issues with the poor plasmid recovery when analyzing larger plasmids with the CMAC pDNA column with 1.4 micrometer channels. To address this issue, we have developed a new CMAC pDNA analytical column with larger 6 micrometer channels. Larger channel size has enabled us to resolve the poor recovery issue for larger pDNA isoforms. The two methods for small and big plasmids were validated on all important parameters including LOD and LOQ, linearity, accuracy, precision, and robustness. All validated parameters comply with the criteria set on the ICH for biological analytical method validation. During the development of the pDNA analytical platform, we encountered another significant challenge. In discussions with a number of clients, the majority have highlighted difficulties in setting up a chromatographic analysis at their site, citing a lack of knowledge and chromatographically educated personnel as a key issue. To address this challenge, we have streamlined the process to make the pDNA chromatographic analysis more straightforward and intuitive for our clients. The first step was to develop new PATfix software that is much more straightforward than conventional HPLC software and easy to use. The software also allows complete control over the PATfix hardware and detectors. We have implemented a comprehensive step-by-step guide for the pDNA platform, ensuring that even those without prior experience can perform analytics with ease. In conclusion, the PATfix pDNA analytical platform provides a comprehensive and robust solution for the accurate characterization and quantification of plasmid DNA across all stages of production. By overcoming the limitation of traditional method, it ensures precise separation of pDNA isoforms and contaminants, making it an essential tool for optimizing pDNA processes. The PATfix platform is accessible to all, even those with limited chromatographic experience, thanks to its user-friendly software and streamlined protocols. This empowers companies to efficiently insource their pDNA production and purification. Thank you for participating in this demonstration. We hope that you gain a better understanding of the PATfix pDNA analytical platform and how it can benefit your manufacturing processes. If you are interested in learning more about the PATfix pDNA analytical platform, please visit our website or contact our sales team for a personal consultation. We also invite you to download and try our PATfix demo software, available on our website. Let's work together to take your pDNA analysis and production to next level.
Well, thank you for a fantastic presentation. Next up, I'll pass over to Ahmet to start his presentation.
Thank you very much, Roshan. So my name is Ahmet Tekin. I'm, as introduced, a senior operational scientist at BioNTech at the site in Marburg, at the microbial manufacturing department, or the Miami project, for establishing a pDNA manufacturing platform. We are one of those customers that Nate and Blaž has mentioned, who had some struggles with our plasmid manufacturing. So we tried out the PATfix and integrated it into our analysis of our steps. To give you a short introduction to who we are, as I mentioned, the microbial manufacturing in Marburg. We are approximately 50 employees. We have different scales for different things we want to accomplish. We have a small scale to manufacture clinical material. we have a large scale for the production of commercial scale plasmids. As far as of now, we only have the COVID-19 vaccine as a commercial product, and our PRIDE project, which is called Process Implementation and Development, in which we try to further develop and make our process better, shortly said. In this presentation, I will go into depth of the alkaline lysis and depth filtration, linearization, and purification, because the transformation, cell banking, and fermentation are not as part of my presentation or as needy. And we don't need the PATfix platform to analyze those steps. Shortly, the PATfix platform helps us to analyze our process. Previously, we only had the UV measurement via our quality control department to check for our content and quality of our product. And because we are a GMP facility, it was a long or a lengthy method, which is GMP validated and takes too long and is not for our benefit. So we reached out to our friends at Bioseparations and Sartorius, and they handed us a solution, the PATfix HPLC, which with a weak anion exchanger, which accomplishes a simple and fast analysis, characterization, calculate our concentrations, and do qualitative and quantitative analysis of our process. In the chromatograph on the right, you see the standard of Sartorius or Bioseparations. In red, you see the open circular and supercoiled standard, and in blue, our self-made linearized pDNA standard, which we use to quantify our product. And in the further detail, which I want to talk about in the alkaline lysis, is what steps or what points in the alkaline lysis we are able to take a closer look at and see how we can make it better. So first off, what we have to see is how can we efficiently get more plasmid out of our yield when we lyse our cells. To see in which points we need to take a closer look at is a convenient mixing equipment or method, because if we rigorously shake our cells during our lysis, we just damage all our plasmid, which is not good. We played around with the suspension buffer volume 10 times, 20 times, and 50 times and checked how we can accomplish a higher yield. Also, we played around with the concentration of sodium hydroxide in our lysis buffer. Our precipitation buffer, we played around a little bit with the final concentration in it, and overall just our buffer volumes and additions, and checked what the yield said. So we had our different concentrations and yields that we tried to check. And in this, you can see that first off, the first peak is all the contaminants that Blaž was talking about that are shown in the that elute from the column. So we had difficulties in PATfix with our product because the UV signal was too weak because of low concentrations of our plasmid due to high volumes of the buffers. So what we did is we changed our sample loop from 100 microliters to 1,000 microliters, which enabled us to just load more sample and get a longer detection of the signal. Also, what we did is we changed the UV flow cell to get a longer pathway or a stronger signal detection. So in the next step, we take a closer look at our ultra and filtration step and to check how to not kill our pDNA. Also in this step, we need to be careful and gentle while we're handling our pDNA or when we put our pDNA on our ultrafiltration system, our TFF system, because if we process it too harshly or put it on with too much pressure or too high flow or not the right or optimized parameters, it's easy to clog the the filter cassette, or we just produce a gel layer on top of it because that's what the pDNA tends to do if we don't have the right parameters, which also happens to us. So we had to try to find the sweet spot of the different pressures and flows and to not clog our filter, and at the same time not put too much shear stress on our pDNA to essentially break it down. In the next step, how we try to optimize our chromatography is we were thinking about, is it possible to just put the anion exchange directly after the depth filtration and not just go through an ultra diafiltration first? So what you see is on the left directly the anion exchanger or the chromatography after the depth filtration, which shows just a very, very low amount of plasmid in elution. And if we just load it directly after the ultra diafiltration, we saw that the plasmid is in the wash. Our problem was that we probably didn't get our right concentrations. So we started to optimize our buffers a little bit around here. The main problem that we had was that our conductivities of the buffers were not right. So the pDNA and RNA concentrations of the product were too high, and we didn't see a differentiation of it. So when we did our anion exchange, we, of course, also tried to upscale it. On the left, you see only 2 fractions, which is basically only supercoiled DNA and no RNA. On the right, you see the chromatograph of the anion exchange after ultra diafiltration, and we see that we only have 1 fraction, and the wash fraction, the open circular, supercoiled, and RNA. So in the next step, what we try to do is we try to load our column of the chromatography only with a low amount of plasmid. So we try to use it with only 10% of the maximum capacity that the column is able to hold. And what we saw is that it actually worked very well after we increased or decreased DNA concentration, or we didn't put too much, how should I put this? We didn't overload our column with DNA. That's actually what you can see in this slide. This is what happens when we just tried for fun to overload our column with about 200% of its capacity, and we see that we don't get any clear peak. It's just a noisy signal that we get throughout all of this, all of the chromatograph. So another crucial step is the linearization that we try to achieve, because as you know, the mRNA-based vaccinations need to be linearized or need to have a linearized plasmid as a starting material. We do this via enzymatic linearization of our pDNA. At first, we started with a 24-hour time course of enzymatic restriction. And we optimized this by just taking samples of our linearization process, just taking samples every hour and check how our linearization steps, how our linearization process is shown. In blue, you can see basically time point 0. In red, you see time point 1, and in green, time point 2. So what we can observe is that the supercoiled version basically changes to open circular first, and after that in the linearized version. So we saw, as I said initially, a partial digestion of the DNA, which shows the open circular peak, and afterwards the complete linearization of the plasmid. In the last optimization point, we have our HIC chromatograph, or the HIC chromatography. What we were able to see is we had a double peak in the HIC chromatogram, which is maybe due to secondary interactions and differences in the conformity, or also what we're able to see is that in an isogradient gradient, it shows no double peaking, but a slight shoulder. So on the left, you see the this so-called twin peaking, what I was talking about. Also, this shows in the PATfix analysis, this twin peaking, when we take a sample out of the HIC chromatograph. So just to wrap this up a little bit, what we were able to achieve with analysis of our process or characterization of our process with the PATfix platform is we were able to see lots of contaminants in our lysis, but also able to see our DNA concentration, and we were able to do a qualitative analysis of the plasmid during the lysis. We were able to see our partial removal of RNA and contaminants, or concentration of DNA, and also that our buffer exchange for chromatography, that it works better if you do it before we load our DNA on the anion exchange chromatography. In the AAX, we see itself a complete removal of RNA and the partial separation of the isoforms, because as you know, before we go to linearization, what we want is a supercoiled isoform of the DNA. What we also see is that the linearization is successful after a shorter amount of time, and not the initially started 24 hours of enzymatic restriction. We were able to see that in the HIC, we remove the endotoxin, which, as Blaž was mentioning, has a high similarity to DNA. So we are basically purifying it along with our DNA, but in the HIC, we are able to remove this. And so far, we were able to increase our plasmid yield 8-fold after our first process optimization. And we did this thanks to the PATfix platform, which is able to visualize and easily analyze our samples in a fast, it's basically really fast and easy to analyze our samples without sending it to our GMP validated method QC quality control department, which takes a long time to send us our results back. And with this is basically my end. I will thank you for listening to my presentation, my co-workers who helped me with the presentation and mainly do the PATfix analysis, all of our operational scientists and development team, our production personnel, of course, Sartorius and Bioseparations for helping us with the PATfix platform, and Copic for helping us with the development. And this is it from our side.
Well, thank you for some great presentations. Before we move on to our Q&A, we would just like to ask the audience a quick question, which should pop up on your screen now. Great, thank you for your input. We're now going to start our Q&A. Please do keep your questions coming in. We'll get to as many as we can just now, and any others we can answer by email. So, Blaž and Ahmet, if you'd like to join me back on camera.
Can you see me?
I don't seem to be able to see you just yet. Well, if you can hear me okay, I can still kick off with the first question, which is for both of you. What are the biggest challenges in the pDNA purification field? And can I get both of your views on how it's going to evolve in the coming years? Blaž, if we start with you first, maybe.
Oh, sure. Thank you, Roshi. The biggest challenges, I would say, as the industry moves forward and really gets on board with the gene therapy and mRNA vaccines even more in the future, the scale and scope of pDNA production required to satisfy that mRNA and, for instance, AAV production is going to increase hand in hand. And I would say on the process side, probably the biggest challenge is going to be scaling up in volume, which is something that Sartorius is already addressing. If you're interested, let us know. And on the scope, I would say that while the pDNA lysis, so E. coli lysis, pDNA purification seems simple enough on the small scale, on the large scale, it can get very tricky and complicated, especially when you're chasing GMP grade quality.
Fantastic, thank you. And Ahmet, would you like to weigh in on this question?
Yeah, I think Blaž already filled this out pretty well. I think the amount of pDNA that is needed, or the demand for pDNA will be, or is in such a high volume that has not been addressed so far. Also in GMP grade, it's pretty easy to manufacture, I think, pDNA in a lab scale, but not in a commercial scale as we might need it now in the next few years, I think.
Fantastic, thank you both. Next up, I have a question here for you, Blaž. What is the pDNA size limit for analysis using PATfix?
Okay, that's a good one. So the pDNA platform itself comes with two methods. One is for smaller plasmids and one is for larger plasmids. And the cutoff between the two is around, I would say, 8-ish kilobase pair of plasmid size. And as far as size on the upper side, we haven't noticed, we haven't encountered any plasmids large enough that would pose us problems. So we've tested over 20 kb and it still worked as it should. So we don't know yet is the answer.
Fantastic, thank you. And staying with you, Blaž, I have another one for you here. Can I implement this method on a different analytical Chrome system?
Yes, you actually can. Now, my counter question to that is developing the process is usually hard enough. So what we offer with the pDNA platform is basically a one-stop shop solution instead of going into it yourself, because there's a lot of moving components going into development of this analytical method.
Absolutely, thank you. Ahmet, coming to you. Usually the HIC purification is done directly after the AEX. So why are you doing it after the linearization?
Yes, thank you. So usually, yes, the two chromatography methods are used one after another, but we try to elegantly get rid of our restriction enzyme after linearization by just putting the HIC chromatography after the linearization process. So it's just an elegant method to get rid of the enzyme.
Great answer, thank you. And Blaž, back to you. Why analyze pre-capture samples for pDNA content?
Another good one. So post-capture, the sample is usually quite purified already and it's much easier to handle, but a lot of the gain and optimization of the process can be had actually in the pre-capture steps. So that's why being able to analyze pre-capture step samples is quite valuable.
Great, thank you. And Ahmet, back to you. Does the twin peaking observed in the HIC purification show any impact in plasmid quality?
Actually, it does not. So we only observe this when we do an isocratic gradient of the HIC purification. And we only observe this after we take a sample out of it and put it in the PATfix platform. But we didn't see, as of now, any decrease or increase of quality due to the twin peaking.
Great, thank you. Another question here that says, excellent presentation, thank you. Could you comment on what the impact is of pDNA isoforms on cell and gene therapies? I don't know, Ahmet or Blaj, if you'd like to weigh in on this one first.
This one is, to an extent, I would say it's still undecided. I don't think the regulators have completely set up their mind on this, but usually better purity and better control over your process is always better than less. And open circular plasmid creation is usually, quite a lot of it is a consequence of processing the pDNA where it's usually formed.
Fantastic, thank you. And Ahmet, would you like to weigh in on this one?
And yeah, I think I'm also not 100% sure on this, but I think also the supercoiled forms are favorable in terms of further processing of the pDNA. But I might have to check in on this.
Fantastic, thank you. And just as a reminder to our audience, do keep your questions coming in. And if they are for a specific speaker, you can add that in the question if you'd like me to direct it to someone in particular. Great. Next up, how did you reduce RNA contamination after lysis? And Ahmet, I think this is for you.
Yeah, I think so. So we observed it's pretty easy to reduce RNA contaminations after lysis by using the anion exchange chromatography. After analyzing our product after the anion exchange, we see that a lot or almost all of the RNA is basically gone.
Great, thank you. And another question here. Can pDNA manufacturing be improved in terms of speed to meet requirements in the event of a disease outbreak? Who would like to take this one?
I think this one's for Ahmet.
I think in terms of speed, you can only improve the manufacturing speed of pDNA to a specific point, right? Because at first you have the fermentation, which is the E. coli, which is the living system basically, and by just giving it the time to produce the pDNA by itself, and after this, all the purifications and further steps, that's not a long time. I think the longest time that it takes is then the releasing of said product, right? All the quality controls that need to be right. And this is then everything that takes a lot of time, or more time than the manufacturing itself.
Great, thank you. And Ahmet, I have another question for you here. Do you detect any recombination in your plasmids? And if so, how do you address this?
So we only detected recombinations of our plasmids after the transformation process, and then it's hard to get rid of those recombinations during the purification. So basically, what you do is you have to start with the right plasmid in the beginning. And if we start with the right plasmid, what we didn't see any further recombinations. And if we do, I think we saw some slight different peaks in the chromatographs, but also we're able to separate it in the chromatographs.
Fantastic, thank you. Now bear with me as I seem to have a two-parter question here, which says, do you have any type of problem with the TFF-DF in the process for changing the buffer of HIC? Oh, hang on. Any problem for changing the buffer of HIC for water at the end of the process? So problems like loss of the pDNA or breaks in the pDNA?
As far as of now, we didn't see any problems with the diafiltration of the HIC buffer. But I mean, we don't exchange it for water, we exchange it for another buffer in the end. So maybe that's that might be one of the issues, but we don't see any difficulties with this.
Great, thank you. And another question here. RNA contamination reduces AEX binding capacity. So how did you address or resolve this?
So we decided to not use the column to a full capacity. So by just approximately using 10 to 50% of the column's capacity with our pDNA, we try to not overload it to not reduce the binding capacity of the column with the RNA.
Fantastic, thank you. Well, thank you for answering those questions. I'm afraid that's all we've got time for today. So any questions we didn't get to, we will reply by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar. The link to this can be found in the Session Resources tab on the left-hand side of the screen or in the on-demand email. So all that's left is to thank Blaj and Ahmet once more for some great presentations, and thank you to the audience for watching. We hope you'll join us in the next session today, which will be covering enhancing mRNA production with advanced analytics. This is starting in just a few minutes, and you can find it by returning to the event lobby. Thank you.
Thank you.
Thank you. Bye.
Bye.